Hello ladies and gentlemen, my name is Dylan Hamilton from C.A.R. Entertainment, and today we're going to be moving on to cells in our revision guide of AS1 biology. We're going to start off by talking about the eukaryotic animal cell. This is going to fall just after microscopes, so I do recommend going to watch the microscopes video, which is the second one we put out in the series, before watching this. Basically, you'll see in the top right hand side, that's basically describing what a eukaryotic animal cell consists of, such as a cell surface membrane, cytoplasm, nucleus, the host of membrane systems, and an organelles embedded through it, and you can have detailed view of this through an EM electron microscope known as ultrastructure. So let's get into the video anyway. So the nucleus, we're basically going to go through all the different things you'll find in the cell, all the different organelles. So let's start off with the nucleus. This contains DNA and chromosomes. Cells are not dividing chromosomes as they're not visible in organized chromatin, as organized in chromatin. Chromatin is densely packed. This is heterochromatin, and so it's dark. But the lessly dense chromatin is called euchromatin, and this is lighter. A nucleus will contain one or more nucleoli, and the nucleolus will be darker than the heterochromatin. So you'll be able to see in the bottom left of the diagram, which makes it a little bit more clearly for you to understand. You'll see the nucleus there is the black blob. The nucleolus synthesizes ribosomal RNA by making ribosomes, and these are going to be found all the way through around, mostly in the Golgi Apparatus. The nucleus is the control center of the cell, with DNA and chromosomes coding for the synthesis of proteins in the cytoplasm. By isolating chromosomes, the rest of the cytoplasm reactions will take place, and their DNA will be protected from damage. The DNA code will be taken from the safety of the nucleus to the cytoplasm, and this is achieved by nuclear pore to the nuclear envelope, and the nuclear envelope has a double membrane with a very narrow space between each membrane. So now we're going to talk about the endoplasmic reticulum, or the ER. So the ER is a membrane system extending throughout the cytoplasm with 3D enclosing sacs called cisternae. The ribosome organelles will be dotted alongside the outside of these membranes, and they can be called the rough endoplasmic reticulum, or the RER, and the other parts which do not have ribosomes attached to them, and this is called the smooth endoplasmic reticulum, and this is the SER. So we're going to abbreviate those and just describe it like that. So the RER provides a scaffolding for protein, and the ER is operating distribution network for proteins. The ER will be joined with a nuclear envelope facilitating the transport of RNA, and the RER is particularly common in cells whose function is to secrete protein. SER roles include the metabolism of lipids, including the synthesis of cholesterol. Again, diagram on the bottom left, you may not be able to see that massively clearly, but it is just sort of a better view of what they look like, because you might be asked to label. Ribosomes are small organelles as small black dots in an EM micrographs found in the cytoplasm attached to the outer surface of the ER, and these are made of protein and ribosomal RNA. They frequently occur in polyribosomes, creating hotspots of protein synthesis, 50% of ribonucleic acid, 50% protein, and ribosomes are the site of enzyme and protein synthesis, which is very, very important. Once again, diagrams, you'll see diagrams throughout here to try to make it very clear of what they will look like. So the Golgi Apparatus, this is quite important because it's a series of curved cisternae with a number of small vesicles entering and leaving the system. Vesicles or vesicles containing newly synthesized protein will pinch off the RER, forming a convex edge of the system. Within these proteins will be a modified and packaged carbohydrate added to form glycoprotein. These vesicles with a finished protein will be pinched off the maturing concave face, and these vesicles will transport the protein within the cell and fuse with the cell surface membrane, releasing the contents outside of the cell. Whenever we go on to talk about protein synthesis later on in a separate video, you'll understand this a lot better. Lysosomes are very tiny vesicles containing something to be destroyed. Whenever they fuse, they form secondary lysosomes which have an important role in phagocytes, and digesting engulfed bacteria is enclosed in the phagosome. Again, if you remember back to our previous video, we talked about immunity. If you're doing A21 this year, then you'll know about lysosomes and their process of phagocytosis. Lysosomes have a relatively thick membrane. Mitochondria now, this is the powerhouse of the cell, and they're present in almost all types of animal cell, and they're in sausage shape, enclosed within a double membrane separated by intermembrane space. The inner membrane will be folded to form cristae, extending into a matrix of mitochondria, giving an inner mitochondrial membrane with a greater surface area, increasing the number of enzymes embedded within this membrane. The mitochondrion is the powerhouse of the cell, like already mentioned, as it is the site of ATP synthesis during aerobic respiration. It's common in cells with high energy requirements such as muscle, as the ATP is needed to be provided. The cristae will tend to be more numerous and more deeply enfolded in these highly active cells. On the microtubules, these are hollow cylinders formed from the protein tubulin. They're found throughout the cytoplasm, occurring within centrioles, forming the spindle fiber essential in nuclear division. When we go on later on in AS1 biology, we'll talk about mitosis and meiosis, and these will be important. In the centrioles, they'll form 9 triplets of microtubules arranged in circular formation, found in the cilia and flagella. You'll just basically need to know where they're found there. Microtubules are part of the cytoskeleton, helping direct the movement of cell organelles. Now to the big one, which is the cell membrane. This is the one they go in most in depth with. So the cell membrane is basically a boundary of the outside world, and it consists of 2 basic components. The phospholipid bilayer, forming the skeleton due to the properties of phospholipid molecules. These phospholipids are hydrophilic. If you remember back to our previous video on lipids, the phospholipids are hydrophilic as they have heads, but the 2 hydrophobic tails and the 2 layers arrange themselves with the hydrophobic tails away from the solution, joining each other, and the heads are mixing with the aqueous solution. Okay, because the tails are hydrophobic and they do not want to be in the water because they're water hating. Hydrophilic heads are, so they will be in the water, or the aqueous medium, I should say. So the cell surface membrane has a hydrophilic head always on the outside and the tails on the inside. That's basically the best way to put it. The protein molecules will be scattered throughout this membrane, and they will be attached peripherally to the bilayer. This is extrinsic, or they'll be integrally embedded into 1 or 2 of the layers. This is intrinsic. Some of these intrinsic proteins extend across the bilayer. Cell membrane part 2. So there's a diagram of what the phospholipid bilayer looks like and the cell membrane. So the structure is described as a fluid mosaic model, as the phospholipid molecules flow sideways, keeping the bilayer arrangement with proteins floated between the phospholipid. The extending phospholipid is a glycolix, and this contains polysaccharides bound to the membrane proteins such as glycoproteins and glycolipids. The carbohydrate will always be in the outside peripheral to the phospholipid bilayer. To basically give you a summary, the phospholipid bilayer is the backbone of the cell surface membrane, giving membrane selectively permeable properties. You'll always hear that there. The cell membrane is selectively permeable. Very, very key. Cholesterol. The purpose of cholesterol is to increase membrane stability, as it restricts the sideways movement of the phospholipid molecules at high temperatures. But low temperatures, cholesterol will help maintain the membrane fluidity, acting as a wedge between the adjacent phospholipid molecules, stopping the adjacent molecules sticking together. So essentially, all cholesterol does is make the membrane less fluid at high temperatures and more fluid at low temperatures. Talking about proteins and cell membrane now. So proteins will provide stability and support each other, anchoring the phospholipid molecules. Proteins will act as enzymes, and the advantage of this is that they are kept in the ideal place in terms of substrate availability and pH, needing to replace left often. If you go watch our enzymes video, then you'll be able to know this in more depth. The enzymes in the membrane will include the membrane of small intestine, which is very important in digestion of disaccharides. Some membrane proteins will act as adhesion sites, as the adjacent cells will be held together and the proteins involved in cell recognition. How do proteins transport across this selectively permeable cell membrane? Basically, they transport by acting as channels, as they create hydrophilic channels, allowing polar molecules bypassing the hydrophobic center of the bilayer. Then there's gated channels. Don't get these mixed up, they are different, as gated channels control the passage of ions into and out of neurons during the nervous conduction. Then we have carriers. Carriers are where basically proteins carry specific ions and molecules across the membrane. Molecules have a charged group, so the ions have to be moved against the concentration gradient, and the carrier protein will change shape, carrying the substance from one side to the other, which is different than the channel, as it just literally moves in and out. There's no protein to help it. The glycolix is where polysaccharides bind to the membrane protein or phospholipid found on the outside of the cell surface membrane. Glycoproteins and glycolipids involved in cell-to-cell recognition allows the cells of similar types to recognize each other, forming tissues. The glycoproteins will act as antigens and then act as receptor sites. This provides sites in the cell surface membrane so particular molecules fit because the receptor sites in specific molecules are concerned. This is important in hormone action and the passage of neurotransmitters between neurons. Basically, C.A.R would very handily just ask you a question, suggest why glycoproteins and glycolipids are involved in cell-to-cell recognition. You'd basically say because they recognize each other and then form tissues, and this is important in hormone action and the passage of neurotransmitters between neurons. Just to finish off, the glycoproteins and glycolipids will form hydrogen bonds, helping to stabilize the membrane, and that is us finished with the eukaryotic animal cell. A very long-winded video. Cell is going to be quite long. It's what I believe is the hardest part of biology in AS1, so hopefully this has cleared it up and it's been a lot easier for you to understand. But anyway, I've been Dylan from C.A.R Entertainment, and I will see you next time.